Right, this is synapses. A synapse is the gap between two neuron cells. Neurons don't actually touch each other. So here we might have, for example, a sensory neuron, and this might be the next neuron, the motor neuron. So there's actually a very small gap. It's about 20 nanometers across, which isn't very big, but it's enough to slow the nerve impulse down, because the nerve impulse is travelling quite quickly along this axon. If it's a myelinated axon, if it's got the fatty sheath around it, at maybe about 400 meters per second. Across the gap, it slows down considerably. Slows the speed of the nerve impulse down to about 15 meters per second. So how does it do it? Well, the nerve impulse has travelled all the way along our axon and it's come to the end. Here's our nerve impulse. And what we have in the end of the neuron is a chemical that we would call neurotransmitter. Which I'm representing with these blobs of green and yellow plasticine. Now, when the impulse reaches the end, these neurotransmitter molecules diffuse out into the gap, and they diffuse across the synapse, and on the other side what we have are receptor molecules, and these have a particular or a specific shape to the neurotransmitter. The neurotransmitter fit into these receptor sites, and that will then cause another nerve impulse to be passed along the motor neuron. So our impulse travels down, neurotransmitter diffuses across the gap, and then continues in the next neuron. Now what then has to happen is that this neurotransmitter must be broken down by the action of enzymes, and it's actually then reabsorbed back into our sensory neuron, and it gets recycled back into neurotransmitter. Okay, now if those molecules of neurotransmitter aren't broken down and they stay in the receptor, this motor neuron will continue to send nerve impulses down here. So if this was, for example, a motor neuron controlling your breathing in and out, it might continually be telling your muscles controlling your breathing to stay contracted, in which case you would suffocate. So it's quite important that these receptor sites don't get blocked off, and that the enzymes that break down these the neurotransmitter are able to work. Now some drugs, I'm going to use a couple of red discs here to represent the drug. A lot of drugs actually will work at the synapse. So for example, what Prozac does is, Prozac is a drug that's a chemical used in preventing conditions like depression. And what Prozac does is it blocks off this neuron so that the neurotransmitter can't get back in, and it stays in the gap, and so it keeps firing these nerve impulses down. And in the case of Prozac, the synapse it blocks is blocking off a neurotransmitter called serotonin, and this is found in the brain, and it's a chemical neurotransmitter we would associate with feelings of happiness. So people with depression often either aren't producing enough serotonin, and so Prozac helps by keeping what serotonin there is in the synapse. It's not getting reabsorbed back in there. Other drugs like ecstasy also have a similar effect in that they can prevent this reuptake of serotonin, and so can lead to feelings of temporary feelings of happiness, although there are obviously unknown effects from the use of ecstasy, in that it's an unregulated, illegal drug, and long-term effects, it's certainly not clear what the effects could be. Any kind of drug that is affecting the action of your brain potentially can have damaging effects. Another side effect of this, if we look at the example of something like a nerve toxin, like for example something like curare, which is found in poison arrowed frogs, those little coloured frogs, the really bright green and black and red and black, yellow black frogs that you get in places like the Amazon. It's a nerve toxin, and what curare does is it will block off these receptors, so that even when the nerve impulses reaches the end, the neurotransmitter is released, it never gets across here, the motor neuron never gets to fire.